Welcome to the final part of this lecture. It again deals with G proteins, a somewhat different kind of G protein. It's the RAS oncogene family, and I will explain how their location is important for their activity and how microscopy was used to learn more about their location and also about ways to interfere with their location, which turns out to be a very viable way to modulate their activity and to come up with interventions. Now, many proteins can be modified with so-called fatty acids. These are hydrophobic lipid tails that make sure that those proteins are recruited to membranes. It can be either the plasma membrane, but can also be the membrane of secretory vesicles. Now, many proteins are modified with these kind of lipid tails. One of those class of proteins is the RAS oncogene family. There are different RAS oncoproteins, HRAS, HRAS, NRAS, and KRAS. And when they are mutated at residue 12, 30, or 61, their GTPase activity is changed. And this mutation causes their constitutive activity, so they're always turned on. And this, of course, is a problem and leads to cancer. Now, these proteins differ quite a lot at their C-terminus, and this C-terminus is also the region where fatty acids can be attached. Now, there are different fatty acid modifications that can occur on proteins, and they differ in their reversibility. So, for instance, we have myristate. This is attached to the N-terminus of a protein. If after the starting methionine a glycine is found, this glycine can be modified with a myristate, and this modification is irreversible. Then we have S-palmitate. It's a slightly different fatty acid that's attached to the protein. It's attached to cysteines, and this modification is reversible. So it can be attached to a cysteine, but it can also be taken off. And then two more that are relevant for this story, farnesyl and geranyl. These are generally also indicated with prenylation. So these prenyl tails can be added to a so-called CAAX box. And this CAAX box you can find at the C-terminus of proteins, as we will see. And these prenylation modifications are also irreversible. And whether this modification is reversible or irreversible has important implications for the location of this protein. And I will explain that later on. Now, if we look at these different RAS isoforms and we look at the amino acids in their C tails, we see that NRAS, HRAS, and KRAS all have this cysteine followed by three other amino acids, and this is a classical CAAX box. So this can be modified with a prenyl fatty acid. And then we see that also HRAS and NRAS have cysteine residues at the C-terminal tail, and these can be modified by palmitoylation, as we will see. Now this is slightly different for KRAS. It doesn't have a cysteine. It only has polybasic residues over here. So this variant can only be prenylated and not palmitoylated. So after protein lipidation, the proteins look like this. They have a fatty acid tail attached to this cysteine, and the three remaining amino acids are cleaved off. Now, as I said, the NRAS and HRAS can be further modified with palmitoyl tails at these cysteines, and KRAS cannot. Now, we can verify what this means for the location of these proteins by making fluorescent protein fusions. So, for instance, you can make a GFP with one of those CAAX boxes and look at the location of this protein. And of course, we can do the same for those NRAS proteins. Now, you can have an idea of, or you can predict the localization of these proteins. As I said, they have fatty acid chains, so they're probably recruited to membranes. And now the question is, which membranes? Now here we see the localization of the CAAX GFP, and here the localization of the GFP tagged with NRAS. And there are quite some differences. There are also some similarities. So in both you see this fluorescent blob near the nucleus. You also see it over here. And in this case, a FRAP experiment is done to look at the mobility of those proteins. So we bleach away a region, and then after a while we take another image, and you can see that within 100 seconds the image looks the same as the original image, which means that those proteins, while they have a lipid chain, can rapidly move between those membranes. Now there's a difference in the localization. So what you see here is this reticular kind of structure. It's ER-like, while here you see mostly, and you have to believe me, this is the plasma membrane, and you hardly see any ER. It's kind of difficult maybe to appreciate from these kind of images, but these are really typical for plasma membrane localization. And of course this is now quite interesting, because both these GFP are tagged with stretches of amino acids that are lipid modified, but you still get quite a different localization. Now, there's an interesting experiment that you can do, because you can take this GFP NRAS, which is located at the plasma membrane as well as at the Golgi, and now you can use an inhibitor. It's this compound, 2-bromopalmitate, and it inhibits specifically the palmitolation. And as we have seen, the palmitolation is reversible. So if we start inhibiting, what will happen with this NRAS molecule that has 3 lipid anchors, when you use the inhibitor, only the prenyl tail will remain. And now the question, of course, is what will happen? How will the localization look like? Now, after treatment with this 2-bromopalmitate, what you will see is again this Golgi localization and this reticular type of localization that was similar to the CAAX localization. So now this NRAS localization with a single prenyl chain starts to look like CAAX. And it's quite different actually, strikingly different from this intact GFP NRAS that localizes to the plasma membrane. And now the question is, how does this happen? Why is this localization so different between those two molecules? Now, one of the potential reasons for the difference in localization is this reversible acylation on the palmitate groups. And so the idea of the researchers was, what would happen if this palmitoyl chain is there forever, when it's always stuck there? And this is a question that they try to address. Now first, let's have a look at the different HRAS, NRAS and KRAS isoforms and how they localize in cells. In this case, we have fusion proteins, and it shows that for HRAS and NRAS, with the reversible palmitolation, you see nice plasma membrane labeling. And next to that, you see also some intracellular labeling, which coincides with the Golgi. So this HRAS and NRAS proteins really localize between Golgi and plasma membrane. Now the KRAS is a slightly different story. The KRAS only has this prenylation, and you see it only has plasma membrane labeling. So they also pursue this localization, but we'll not discuss this here. Now, to look at this reversibility of the palmitoyl group, the researchers made two different variants of this protein, and this was a tremendous effort to synthesize these probes. So they made an NRAS, attached a fluorescent molecule to that, attached the farnesyl group and the palmitoyl group, which can be cleaved, indicated with the scissors, and they made this alternative variant, which has a palmitoyl group that is slightly different. So it's an analog, and you see it's missing one side chain here, and it's no longer cleavable. So they made this variant that has a palmitoyl-like fatty acid chain, but it's not cleavable. And then, because these proteins have a fluorescent probe, they need to be micro-injected into cells, and they looked at the localization. Now, if we look at this natural variant, which can be reversibly palmitoylated, we see nice plasma membrane labeling and Golgi labeling. And quite surprisingly, if you make a very similar molecule with only a very minor change, what you see is no longer this plasma membrane localization, but you mainly see a reticular structure which is reminiscent of ER. So this indeed shows that this reversible palmitoylation is necessary for plasma membrane localization. And then they came up with this model in this cartoon. So what happens with RAS is that it's prenylated and then palmitoylated at the Golgi. So that's why you see the Golgi localization. Then with the secretory pathway, these proteins are brought to the plasma membrane. That's why you see it at the plasma membrane. Now what happens with any of those fatty acylated molecules is that they at some point will diffuse throughout the cell and participate or spend time on all these membranes. Now what happens when these molecules diffuse over the cell is that they encounter this enzyme, the thioesterase, and this thioesterase is capable of cleaving off this palmitoyl group. This will increase the diffusion speed, and these molecules at some point will hit also the Golgi, be palmitoylated again, and then they're trapped on the Golgi, and again via the secretory pathway they will go to the plasma membrane. And this is now the cartoon model of why you see predominantly plasma membrane labeling and Golgi labeling. And so the idea is that this thioesterase is some kind of control mechanism that modifies mislocalized molecules to bring them either to the Golgi or the plasma membrane. Now importantly, RAS, which is an oncogene, and if it's mutated, it's constitutively active. The idea is that it mainly, the activity at the plasma membrane is very important. Now, if you can interfere with this plasma membrane localization, of course, you could also interfere with the oncogenic activity. So knowing more about this system and finding ways to interfere with that could be a nice intervention strategy. So the researchers thought of this experiment where they have a method to make sure that these palmitoyl chains stay on there. So the way to achieve that is to inhibit the degradation of these chains by the thioesterase. So this is the enzyme that cleaves off these molecules. So if you could design an inhibitor that inhibits the APT1, you interfere with this quality control system. So they started to screen for drugs that inhibit this APT1, and they actually came up with one of those inhibitors described in this very nice paper in Nature Chemical Biology. And here's the experiment that they did with this inhibitor. So here you see again NRAS localization at the plasma membrane and partially also at the Golgi. Now when they add the inhibitor of this APT1, what they see after 90 minutes is that this protein, the NRAS, starts to delocalize over all the membranes. And since its activity at the plasma membrane is important, they then argue that this is a nice way to decrease the activity of this oncogene. And to examine that, they did this experiment in which they looked at MDCK cells. So the cells are over here. These are normal cells, and they stained for cadherin, which localized nicely on the cell borders. And then they also looked at this MDCK variant, which has a constitutive active RAS. So these are transformed cells in which RAS is active. And what you see, they have a different phenotype, they look different, and also this cadherin staining at the borders is lost, which is one sign that they undergo EMT. Now, what happens if you add the inhibitor that inhibits the APT1 to these cells? What do they look like? So that's the experiment that they did. And now what you see is that the cells start to look more like the wild type cells, and also that this cadherin staining is rescued to some extent. So you see the outlines of the borders again. And this is now a very good indication that you start to really interfere with RAS activity, and probably you do this by inhibiting or interfering with this localization. So this is now a very nice new potential inhibitor to inhibit RAS oncogenes. So the conclusion from this story is that interfering with the location of an oncogene, so instead of what you would normally do, interfering with the activity, is an important strategy to prevent the activity. So changing the location can be a good strategy instead of inhibiting the activity. And of course, this is quite a new concept in this field. Now, to summarize everything that I've told you, here are some important points. To understand a cell, you really need to observe it. You have to have a look at it. Now, the timing of G proteins and their activity determines the signaling output. So you can have multiple G proteins active at the same time, but the balance between their activity and the timing determines the signaling output. And we've also seen that the location of a G protein determines signaling output. So interfering with G proteins and their location can be a good strategy. So I also made some take-home messages. I would say that cell signaling, signal transduction in cells, is all about where are the molecules, when are they active, with who do they interact, and that microscopy is very vital for studying all these aspects, but also for developing novel concepts in cell biology. So microscopy is a tremendous tool to increase our understanding of molecular processes in cell biology, and then as a side effect of that, this will lead to a better understanding and to improve therapies and therapeutic interventions that will be developed.
And now for something completely different.
Then I want to spend a couple of words on the assignment. So there are several assignments. If you take an assignment that I will supervise, then these few words may be interesting for you. The assignment deals with label-free versus fluorescence methods. We've seen both of these methods in the talk. So there was a label-free method that's called ESIS to look at G protein coupled receptor activity. And there are all kinds of fluorescence methods like probes for heterothermic G proteins that can be used to study GPCR activity. Of course, like any technique, each technique has its advantages and disadvantages, and it's up to you to find out what the advantages and disadvantages are. Now, whatever the method, all these methodologies have been very viable for screening drugs and finding drugs that either activate GPCRs or inactivate them. And these are some of the well-known examples that came out of those drug screening efforts. And both label-free and and technologies with labels have been used over the years to find these. And just to remind you, this is the typical data that is gathered. So what the drug industry aims at is generating basically these kind of curves in which you see something that stimulates the G protein coupled receptor as a function of concentration. So the concentration is plotted over here, and then you need to get some kind of response, and it doesn't really matter what that response is, as long as you can measure something that is specific for the activation of this GPCR. And in this specific example, a modulator or a drug was added at different concentrations, and what you see is that by increasing the concentration of this modulator, you attenuate the response. So this is a good drug to inhibit the response of this agonist. And this is, of course, very useful to find these kind of compounds. And that is what both label-free and methodologies with labels are actually after.